Hi, everyone, and welcome to this inaugural episode of The Abstract. I'm Shahin Zagros, your host, and today I will be joined by a truly remarkable individual. This man, in many ways, is responsible for teaching me the ways of the brain way back when I was a green graduate student embarking on this daunting journey of obtaining a PhD in neuroscience. He was there to take me under his wings and gave me all the support and guidance I could ever ask for. What can I say? He's a rock star of network neuroscience, the poster child of connectomics. Of course, I'm talking about my former thesis supervisor, colleague, and good friend, Dr. Boris Bernhard. Boris, thank you so much for coming on the show. How are you doing, my friend?
Yeah, thank you for having me, Shaheen. It's really a pleasure to be here. I'm doing very well and I'm looking forward to the discussion.
Wonderful. So, Boris, before we dive into the science, I wanted to share with the viewers a philosophical tidbit that you imparted on me many years ago. And it's something that I carry with me that in many ways kind of informs my day-to-day decision making. And it goes as follows. Don't let perfect get in the way of good enough. Okay, so don't let perfect get in the way of good enough. I guess my first question to you is, what is the origin of this golden philosophical nugget? And maybe more importantly, what is its significance to you as a researcher?
So how much time do we have? Because I think we can deliberate about like this phrase for quite a long time. Um,
Let's
the
try
or-
to,
the
yeah.
origins of it is that when I was doing my PhD, I had a lab mate. His name is Luis Concha, fantastic neuroscientist and brain imager, and good friend of mine as well. And I think he was the first person who coined this. And it's at this point, it was a lot about the academic writing process in a way that we are obviously trying to work towards something that is as good as it can be. But then we also have to balance this a little bit with milestones, timelines, maybe also that there's like a little bit of a racing character in academia that we want to publish something on time. So I think this is maybe what summarizes it. And like now as a mentor, it's something that I would also like to pass on to like some of the trainees, because some of them, they may sometimes want to achieve a little bit too much, especially at the beginning of their academic journey. So they are very ambitious for their first project. So in a way, for me, determining or like co-designing their project, we try to have some incremental ways to make things better over time, make things better with iteration, and also have a little bit of a trajectory in how our project should be developed, so that maybe not the first project doesn't need necessarily to be the most grandiose transformative project, but maybe this comes towards the end of the PhD or towards the end of some series of analyses. So that's maybe a little bit what I'm also trying to communicate with my trainees and with my colleagues and with my friends. And I also have to live by this, because sometimes having formal deadlines and having intermediary milestones and deliverables is then good, because this can also sometimes help us to change directions, make things better. And I think the peer review process in a way is like also one example of this, in a way that we try to submit fine work, but there's always going to be some feedback from colleagues, from peers, that also make our work better. So even if we haven't submitted the best work at the beginning, by the end of this process, the work will definitely be better. So I think overall, this is a little bit maybe what summarizes why I think this claim is still quite valid.
Yeah. Yeah. And I mean, practically, it also makes sense because perfection is kind of this unattainable, unachievable goal. But as long as like your work meets well-defined benchmarks, well-defined standards, like you said, don't let perfect get in the way of good enough, because you'll be working until from dusk till dawn, and you'll still be working. But so that
I think if I can just answer.
Absolutely.
So I think it doesn't mean that one should always go for the low hanging fruit, right?
No.
I think it's like a fine balance, as you said, between really striving for something that's new, that's better, like not necessarily salami slicing, but finding like this perfect thing, or like almost like the sweet spot in a way between something that is impactful enough but that still remains feasible and that can also be done in a finite amount of time.
Yeah. Yeah. So, I mean, you probably know this, Boris, but this podcast owes its name to the scientific abstract, and that generally contains four key components: objective, methods, results or findings, and significance. And today, specifically, we'll be talking about a paper that your lab published recently in Nature Comms, Nature Communications, by the title of Multimodal Gradients Unify Local and Global Cortical Organization. Now, there are many interesting layers in this paper, but one thing I'd like to point out right off the bat is that the first author of this paper, Yizhu Wang, you were just telling me before we started recording, just successfully passed his doctoral thesis defense. So, Yeju, or should I say Dr. Wang, if you're watching this, congratulations to you, my friend. Welcome to the club, and I wish you all the best in your future endeavors. Now, let's start off with a very basic question, Boris. What was the main objective in this study? What were you guys interested in achieving?
Yeah, thanks for highlighting our work in your podcast. We appreciate this a lot. And yeah, so this is work that's been led by Yizhu. He started this a few years ago. It was a very ambitious project, and it's also embedded in a larger initiative between McGill and the Forschungszentrum Jülich. It's called HIBAR, Helmholtz International Big Brain Learning Analytics Laboratory. So it's a multi-million dollar German-Canadian collaborative project, but also involving a lot of international collaborators and friends. So it's really a team effort that Yizhu has pulled off there. So he deserves all the credits for this. And this study is a little bit embedded in a current debate on both conceptually but also analytically in how can we analyze and understand the brain. And at the moment, there's maybe we can talk about this in more detail a little bit later, but there's basically two camps in a way that try to see what are the fundamental units or what are the basic axes of how the brain is spatially organized. On the one hand, we have a lot of folks, and this is basically even classic neuroanatomy, that have tried to identify different cortical areas, parcellated the brain into discrete units, and build network models based on the interplay of these different units. Then there's like a different alternative or complementary viewpoint on the brain, that the brain is presumably organized along different dimensions. And we refer to these dimensions sometimes as gradients, so more continuous spatial patterns in which brain function, but also microstructure can be organized. And I think from the outset in this study, what Yizhu was hoping to do, and I think he also succeeded in doing, is trying to maybe find a way in which these two sometimes not conflicting, but at least, coexisting ways of how people have dealt with the brain can maybe coexist very friendly, very peacefully, and be integrated and consolidated. So I think this was the main objective. And then obviously, beyond that is maybe to try to see how more local characteristics of the brain can give rise to integrated brain function at large.
Okay. Like any good neuroimaging study, which this project fundamentally is, everything starts with proper data acquisition. So the paper mentions precision neuroimaging, or PNI. Could you maybe in broad strokes explain what precision neuroimaging is, and maybe more specifically what type of neuroimaging modalities were acquired in the context of this project?
Yeah, so Thank you for the question. Precision neuroimaging is a term that has possibly emerged, I'd say like 5, maybe even more years back. And this is basically the prioritization of high quality data, repeated scans in maybe not such a large group of individuals. So we're trying to really profile the individual subject with precision instead of maybe using suboptimal or just very short standard sequences in larger group of subjects. So in a way, we allocate the scan time that we have more into characterizing how an individual's brain is organized. So this is precision imaging at large, and there's like multiple different groups that have advocated for this. The Midnight Scan Club. There's a lot of really cool work from the United States about this, trying to really harness repeated scans. Also, the group of Randy Buckner has done really spectacular work in this. In the data that we have been using for our study, primarily we have acquired a precision neuroimaging data set in-house. So that's at the Montreal Neurological Institute and at the McConnell Brain Imaging Centre, where we installed a 7 Tesla MRI scanner a few years ago. So 7T MRI, for those people who don't know, I always say it's like the iPhone 15 of MRI scanners. So it obviously gives you crisper and sharper images of the brain because the field strength and therefore the signal to noise ratio is just higher. So for Yiju's project, the primary analysis was done in 10 individuals, 10 healthy adults, so young adults, and each of them were scanned for 3 sessions, and each session was about an hour and a half of scanning. So we have 4.5 hours of scanning available for every individual, and then we have this in 10 people.
Yeah. And so the paper mentions that I believe QT1 relaxometry was performed, structural, anatomical data acquisition, as well as resting state functional MRI. So I believe those were the 3 primary modalities that were used in the context of this particular study.
Yes. So one can think about an MRI scanner. That's again like a little bit of an analogy that I sometimes use. There's like this device, I don't know whether it exists in North America, it's called Thermomix. So this is like a little bit of a low-key computer in a way, but it can load different cooking programs.
Okay.
For an MRI scanner, it's a little bit like this. The only limiting factor is time, and you can load different sequences on the scanner, and each sequence gives you a different aspect of the brain. So say you can have one sequence that basically measures brain anatomy. Then you have one sequence that measures brain function. And then you can also measure brain water diffusion. So, and these 3 sequences that you mentioned, so the QT1, that's a quantitative T1 relaxometry sequence that gives you a good differentiation between gray matter, white matter, and CSF. And even in the gray matter, the different intensities, so the different pixel values, they are also sensitive to differences in myelin content of the cortex, but primarily of the cortex. So this is a more myelin sensitive sequence. So just to give an example, on the 7T, the sequence that we used, it's an MP2 range sequence. This is 500 micron isovoxels, so 0.5 millimeter in each dimension, and it takes around 12 minutes to acquire. The diffusion weighted imaging sequence, so sometimes people also call these sequences DTI, but this should possibly be discouraged because it's much more than DTI. So this is a sequence that's sensitive to water diffusion, and water diffusion is fascinating in the human brain because the water cannot diffuse so freely when it hits fat. And myelinated fibers, they are surrounded, so the myelin is fat basically. So the water follows the path of the fiber bundles, presumably. So it's a sequence that we can ultimately use to track axons in the brain. And then the resting state fMRI or the functional MRI. So this is basically a sequence that's sensitive to blood flow changes after neural activation in different brain areas. So hemodynamic fluctuations in the brain. And this can be done when people do a task, or when people just look at a fixation cross, or when people watch a movie or something like that. And in this study, we had people do multiple tasks. I think we'll get to that later, but primarily we also used a very widespread paradigm. It's called resting state fMRI, where people just look at a fixation cross while they lie in the scanner for 6-7 minutes, and they think about nothing in particular. And the interesting thing about these resting state fMRI sequences is, even if they don't do any task in particular, we can still get information on networks that are also engaged in specific tasks. So even if the brain doesn't do an externally oriented task or just looks at the fixation cross, we still can gather a lot of information on functional architecture from the signals and their interactions.
Yeah, resting state fMRI reminds me a little bit of Hubble deep space. Now, I'm not an astrophysicist, but my understanding is that back in the days, it was thought that you needed to look for specific, targets in the night sky to see, stars teeming with abundance. But as it turns out, you can just take your telescope and just point it in just about any direction, and you're bound to see just like a sky that's teeming with stars. And it's kind of reminiscent of this resting state fMRI paradigm, where you could be doing nothing, and yet there's like parts of the brain that are constantly in functional communication with one another, a network that is largely known as the default mode network, or DMN. So you touched up on this a little bit, but an overarching theme in the paper pertains to connectomes and gradients. Maybe tell us a little bit about like what a connectivity matrix, a connectome, is, and how it's used to derive gradients.
Sure. So a connectome, that's a term that's also coined by someone called Olaf Sporns, like one of the giants in the field of connectomics. And that's basically a systematic description of whole brain connectivity. And initially, these connectomes, they were actually derived from track tracing studies. So and now, but in the humans, we use in vivo imaging for this primarily. So some of the modalities that were mentioned before. So one can get a connectome based on structurally sensitive contrasts, based on diffusion contrast, and based on functional contrast. So there are multiple different connectomes in the brain. It depends on how we generate them. When we look at functional MRI or resting state fMRI, what we have is, at the beginning, we have whole brain hemodynamic fluctuations. So all brain regions, they fluctuate. So even at rest. So no brain region is entirely passive. There's always going to be ebbs and flows as we move over the cortex and even in subcortical areas. And what's interesting about these ebbs and flows is that they are not random, but they are coordinated. So you mentioned, for example, the default mode network, which is like one set of brain regions. So medial prefrontal, medial parietal, lateral temporal, angular gyrus, like these kinds of regions, mesial temporal, that on average, they co-activate and they co-deactivate at rest, but also during tasks. So this is how these networks are defined. So this is with function. So we basically correlate time series of voxels, so units in which we measure the brain, and here are hemodynamic fluctuations. And then we get a region by region matrix, which quantifies the correlation between all the different brain areas. And here we interpret each significant, or the high correlations, as connections. So it's basically a region by region matrix at the end that systematically summarizes all the hemodynamic correlations between different brain regions. That would be a functional connectome. For diffusion MRI, the other technique that's also often used, we use tractography. So this is basically a computational technique to follow the water diffusion in tissue to then reconstruct putative fiber bundles. And these fiber bundles then these are not really axons, it's really approximations of it derived from water. So this may be important to say. But overall, they align with how the axonal bundles, or where they go between different brain regions. So from diffusion, we can also then get such a matrix, a region by region matrix. And in this case, it indexes structure connectivity. So the diffusion MRI would give us a structural connectome, in contrast to the resting state fMRI that gives us a functional connectome. And there's other different ways with which we can derive interregional associations. One technique that we have been using in this study, and that also many other folks in the community have been using, is like more of a way to look at covariations of structural measures. And here we used a technique that's called microstructural profile covariance, or MPC. So this is again a different connectome, and here we are basically indexing the similarity of myelin profiles between different cortical areas. So we would have basically 3 different ways in which we can quantify interregional similarity. One based on hemodynamic time series similarity, this would be the functional connectome. One with diffusion tractography, to basically reachability of fiber tracks between areas, and then the other one would be the similarity in their myelin profiles. And usually regions that also have similar myelin profiles, they have similar microstructure, presumably they are part of similar of a similar network, or they might have a similar developmental origin. And you asked about gradients. So all these 3 techniques, so that you can then imagine that you have 3 different matrices, right? Region by region. So this is obviously very complicated. It's very high dimensional. We have, say, if we have hundreds of regions or thousands of regions, then this is kind of a dimensionality that's very difficult to analyze. So what we want to do as network neuroscientists, but also as, when trying to map how the brain might be spatially organized, is we want to bring these matrix data back onto the brain. And for this, we use usually some type of dimensionality reduction, which basically compresses the matrix information into spatial maps. And each of the spatial map describes the variance in each of the matrices. And these spatial maps, they often relatively smooth and they're relatively gradual, and that's why people refer to them often as gradients. So we can get, again, if we have our 3 different matrices, so the functional connectome, the structural connectome, as well as the MPC, we can get a series of spatial maps as principal components from each of them, and then represent variations in them spatially. And these principal components, or eigenvectors, they're usually also ordered in the degree of variance that they explain. So the first one always explains the most variance, the second most, and so on and so forth.
Yeah, that's fascinating. But you guys didn't stop there. You, in the paper, it mentions how you essentially took these 3 modality specific connectomes and gradients, and you kind of like coalesced them. You integrated them into what you call a multimodal gradient profile. So how did this integration, does this integration work?
So this is relatively straightforward in that case. We concatenated the matrices, if I remember correctly. We normalized them so that each of them is weighted equally in the overall process, and then we basically derived a principal component from the combination of them. So with this, we could synergistically represent overall similarity and dissimilarity of different brain areas according to this axis. And it was interesting in that case is that this principal axis also recapitulated at least our foundational understanding of how we think the human brain is organized, and that it recapitulated a lot of a differentiation between more sensory and motor systems from more those that are more higher order, more associative, more engaged in internally oriented cognition. So we could really find a gradient, a space that that could give us a differentiation between externally and internally oriented systems based on these purely data-driven techniques.
Yeah. And so you basically assessed, how shall I say, brain heterogeneity using these metrics known as interareal and intra-areal dissimilarity. So you're looking at, the uniqueness or the commonness of different parts of the brain, and you use the specific brain atlas called the Jülich Brain Atlas, which separates or parcellates, as we say, the brain into 228 different regions. So how was this assessment of brain dissimilarity performed?
Yeah, so these gradients, when we derived them using these computational whole brain techniques, they basically gave us maps in which we could characterize the whole brain. Every cortical point could then be represented as a vector of these. I think we had 5 by 3, so 15 different gradients. So we could profile every region as a 15-dimensional vector in a way that gives us in some way also a uniqueness profile of each of these areas. So some areas might be more specific than others with respect to these different gradients. This is important in the sense that every gradient, while every gradient itself is a bit more smooth, the combination of them at a specific point can generate specificity, which is a little bit so to give context, this is a little bit the way with which you can then still generate functional specificity from smooth gradients. And functional specificity is one of the characteristics of different brain areas. And this is usually an argument from classical neuroanatomists to say that different areas in the brain exist because they have very specific functional roles. So for us, representing each cortical point with a combination of these 15 smooth gradients could then still recover some aspects of areal cortical organization. And in this case, we used a cytoarchitectural defined atlas to show this, namely the Jülich Brain Atlas, which, as you said, divides the brain into 228 really gold standard, as if you want to say it this way, gold standard types of cortical areas. So then we could basically assign a profile of 15 gradients, so the values on each of the gradients to each of these areas. And then we could see in how far are different areas similar or different with respect to these 15-dimensional profiles. And this was basically one of the main findings of the study, in that we could then see that some areas, they may actually not be so different with respect to these, large-scale gradient motifs, whereas others seem to have a more distinctive profile with respect to these 15-dimensional gradient vectors. So we could observe that specifically like sensory and motor areas, they seem to be more specific, whereas regions that are more involved in, so these more specialized, areas that are involved in sensory and motor function, they also develop early, in human development. They are more myelinated, so there's maybe less potential for plasticity and for functional flexibility. So these areas seem to have like a more specific, a more distinctive whole brain gradient motif, whereas other regions that are more integrative, more heteromodal, more paralimbic, they seem to have a less distinctive. So they seem to be also, and this is maybe in line with their presumably more integrative functional roles and their more wide-reaching connectivity profile in the brain.
Yeah, it's truly fascinating, Boris. How were the tasks we mentioned the task fMRI? How were the tasks used to examine functional diversity?
Yeah, so all the gradients that I mentioned before that were ultimately used to generate the profiles, they all came from task-free imaging. So as I said, structural MRI, diffusion tractography, and resting state fMRI connectivity. So no direct task was administered. But because our precision neuroimaging data set also had the patient or the participants do lots of tasks, so we had them in the scanner for quite a while, right? So we also ran like a lot of tasks on them, including some of the tasks that you developed, actually. So relational memory paradigms, but also movie watching paradigms. I think we also had a language task administered. I'm not sure whether this is part of the paper. So our participants, in addition to these task-free sequences that they did at every session, so in each of the 3 visits, they also did tasks. So, and the tasks had some interesting features in that they had matched structure and everything, but you might talk about this maybe in a different podcast session. So what we then did is basically we derived connectivity patterns during each of the tasks. So we did something similar as we did for the resting state fMRI acquisition, but here we did it during the different task context. And then we basically just looked at in how far do the connectivity patterns change from one task to the next. So we basically quantified variations, if you want, in connectivity across these different functional states. This we also mapped back on the cortical surface. So we could map a little bit in which region was most diverse across the task and which region was most consistent across the task. And interestingly, we could find a pretty interesting and consistent relationship between how different the regions were with respect to these intrinsic gradients and how diverse the regions were with respect to tasks. So the more distinct regions, they were less diverse across the tasks and vice versa. And these more flexible, maybe less distinctive microstructurally regions, they seem to be, more diverse across the different functional contexts.
You guys didn't stop there, though. Another very interesting layer that this paper discusses is the fact that you ran kind of validation schemes to make sure that these results were robust. So you leveraged independent data sets and other brain parcellation schemes. So maybe we could finish off with this as far as the methods section is concerned. Talk us through some of these validation schemes that were run.
Yeah, sure. So just as a motivation, when one does neuroscience experiments, one always wants to ensure that findings are robust, findings are replicable, findings are valid. And with this, you can throw some stuff maybe at your analyses and then just try to see whether the results hold. And this is now quite possible because we have a lot of computational power, so we can basically also rerun different pipelines another time and just see whether things hold. Or we can use a different data set just to see whether things are replicable. And in this case, we took advantage of some other in-house data that we had, some open science data sets such as the HCP that I think was used in this work. And so we could basically use, an independent 7T data set, an independent 3T data set, so an iPhone 8 MRI cohort. And we could also cross-validate our findings using a different parcellation scheme in this case. So the initial findings were based on the Jülich brain atlas. And we cross-validated it using the Glasser atlas, which is also a very widely used multimodal parcellation that has been developed by the HCP consortium. So 360 areas instead of 228.
Okay. You touched up on this a little bit, but maybe if in general, you can kind of summarize the overall findings in this study.
Yeah. I realized that I kind of did maybe an introduction, results, methods order. So I mean, first of all, I think for us, it was also one of the first. So this is maybe not what's written in the paper, but maybe just for us. So it was like one of the first demonstrations in the lab that we could really, produce good work with 70 imaging that were collected in-house. So for us, it was like a really important internal application of the 70, which we started scanning in the pandemic. So it was very adventurous. You were there for some of the scans back then. So and we were really pleased with seeing also the data quality coming out of the scanner, despite a lot of piloting needed at the beginning. So the first overall findings were that we could recapitulate the overall sensory fugal organization of the brain. So really trying to differentiate more between sensory and motor systems that are interacting with the here and now, with the outside world, towards these more transmodal association areas. And here we could show this robustly across different modalities. I think compared to some of the work that was previously done, where it was done based on single modalities, here we could really see that this organizational pattern also emerges maybe as some overarching spatial organizational axis in the human brain. So this is really one of the key findings, I'd say. The second one is possibly that we could kind of beyond that kind of see how there's also a similar yet not identical axis of distinctiveness, of integration of different brain areas into the large scale brain organization, where again, maybe the more sensory and motor areas, they are more distinctive, whereas these more fugal, if you want, or more transmodal paralimbic regions, they seem to be less distinctive. And so we have on the one hand, we have one gradient that is kind of giving us maybe an overall cortical hierarchy, but then that also relates to in how far regions are specialized versus integrated into the large scale cortical tapestry. So, and then the second one is that we could cross-validate these findings, in this case, that we're coming from intrinsic features of the brain with more contextual, task-related brain changes in brain connectivity. And we could also see, again, a relatively consistent relationship between how specialized a region is and how much its connectivity pattern changes across different functional contexts. And then thirdly, I'd say that that these findings, although they were initially derived on a small set of very densely investigated individuals, we could also replicate them using another 70 data set that is coming from like a benchmark human neuroimaging initiative, in this case HCP, but also lower field strength open science initiatives such as MICA.
Fascinating stuff, Boris. Maybe one last question as far as the significance of this project goes. What potential future directions for research does this study open up?
So this work is obviously embedded within a more foundational context. So this is healthy young adults, using benchmark data sets, using gold standard parcellations. But for us, I think the next steps are, and like some folks are already working on these things or applying similar methods to other questions. So because in the lab, we also are interested in brain development. So seeing how these principal axes and these principles of structure-function relationships may change across development, I think this could be, a next good step, as well as applying some of these methods to populations with atypical brain development, epilepsy, autism, and maybe also looking at it in the context of lifespan changes and plasticity.
All right, good stuff. Things to look forward to. Um, Before I let you go, Boris, we have a question from one of our members. So I'm just gonna play the clip right now, and then you can reply directly.
Hi, Dr. Bernhard. I'm Yadgar, and my question is if you could scan any historical figure's brain, who would it be and what would you expect to find? Thank you.
I would maybe not scan the brain of one famous single individual, but if I had like a little fairy that would make a wish come true for me, then obviously what one wants to do ultimately is to really track lifespan changes in individuals, and this can be anyone. So ideally, one would have a type of precision lifespan assessment of from birth until late in life, to really look at how the brain dynamically reorganizes as we get older. I know Russ Poldrack. At one point, there's one that's called my Connectome, and I think he scanned himself multiple times throughout a year. But if we had this from a real lifespan perspective, ideally with not just one person but multiple, I think this would be quite fantastic.
Wonderful. Well, Boris, thank you very much. This has been a truly enriching deconstruction of Jeju's paper. Um, do you have any parting words before I let you go?
No, thanks for having me on the show. And I think it's really important, this type of translation of our research finding. For us, it's always challenging to sometimes also talk about things because we're like in the midst of analysis. We use a lot of jargon. I hope I didn't do too badly. But yeah, I think it's good training for everyone to really try to make the work more accessible to the public. So kudos on the initiative.
Well, I appreciate it, Boris, and I really hope that in the near future we get to do it again. I'll be sure to follow your research with a keen eye. And once again, it's been a pleasure. It's been an honor to have you on the show. I wish you all the best, and take care of yourself.
Yeah, you too. Thank you.
All right.